Welcome to all that have joined so far. We're going to be starting in approximately 2 minutes. Thank you to all who have joined us so far today. We're going to be starting in about 1 minute. Welcome. Good morning, good afternoon, or good evening, wherever you may be joining us from. I'm Heather Snyder from the Alzheimer's Association, and it's truly a pleasure to have you joining us today. Today is a webinar that is convened and hosted by the Nutrition, Metabolism, and Dementia Professional Interest Area of iStart. And it's never been a better time to join iStart than today. You can find out more information from the link that's on the bottom of your page. And of course, you can follow the Nutrition, Metabolism, and Dementia Professional Interest Area on Twitter by using the hashtag NutritionPIA. And with that, just a few notes in terms of the overall housekeeping, although I think we've all been on Zoom so many hours and days over the last several months that this should all be second nature now. Make sure you have a solid internet connection. You are in webinar mode, so you are both your video and your audio is muted. We look forward to having an engaging question and answer and discussion period. And on your screen, you'll have the Q&A function. You can submit your questions or comments. If necessary, you can also note that you have a comment you'd like to make live, and we'll do our best to try to take those as well. And with that, I am pleased to introduce today's moderator, Dr. Cecilia Semiere from INSERM. Dr. Semiere is the tenured researcher who works on the Bordeaux Population Health Research Center, and she also serves as the co-chair for the Nutrition, Metabolism, and Dementia Professional Interest Area. Dr. Semiere?
Thanks, Heather. Bonjour, good morning, everyone, or good evening. This is my great pleasure and honor to moderate this second seminar of our web seminar series from the PI Nutrition. A couple of weeks ago, we heard Nikos Karmis discussing recent multi-domain clinical trials for dementia prevention, which was extremely interesting. And today we have the pleasure to hear my co-chair at the Nutrition PIA, Hussein Yassine. Hussein is Associate Professor of Medicine at Keck School of Medicine of University of Southern California. His lab studies interactions between nutrition and diet, lipid metabolism, and APOE genotype in relation to Alzheimer's disease and cognitive aging, with a cross-disciplinary approach from basic sciences to clinical studies. Hussein's talk for today is simply entitled APOE4, Diet and the Brain, but it's a complex question, as you will see. Hussein, the virtual space is yours.
Thank you, Heather, and thank you, Cecilia, for the kind introduction. I'm setting up my presentation as we speak. It's a pleasure to be here, and I hope that I would be able to add to your knowledge on the importance of APOE and how it affects the response to a dietary intervention. All right, so let me just start from the beginning. Okay. Um, so in the next 35 minutes or so, I'm going to talk about some studies that we have done and summarize our studies in relationship to other people's studies on APOE4 diet and the brain. We'll have a good discussion afterwards, and I hope to engage you in this presentation. I have no conflict of interest. I will talk briefly about the basic science of APOE4 pathology and Alzheimer's disease, focusing on aggregation. We'll talk a little bit about recent advances in APOE4, the blood-brain barrier and brain nutrient transport, APOE4 omega-3 metabolism, and get some insights from recent clinical trials. If you would like more information about the content of this presentation, we wrote a detailed review on the interaction between the diet, APOE, brain aging, and Alzheimer's disease in Frontiers of Aging Neuroscience in May of 2020. So this presentation, more or less, is present on that review, and you can refer to that review for more information. At the heart of this talk is going to be an understanding of the basic pathophysiology that allows APOE4 to change how our brains age. So in a nutshell, we study APOE4 because it's by far the most common genetic polymorphism that increases the risk of Alzheimer's disease. And as you could see in this review, the frequency of APOE4 can go anywhere from 5 to almost 40% of the population. That's quite common. However, it's important to understand that the risk of Alzheimer's disease differs by ethnicity. Caucasians and Japanese have a much higher risk of AD if they are carrying the APOE4 allele than African Americans and Hispanic. And that's why it's important to factor in ethnicity when we consider the APOE4 risk on Alzheimer's disease. Fundamentally speaking, although it's only a single nucleotide polymorphism, that has all the implications in creating a system that predisposes the brain to age differently. If you were to remember one thing about APOE4 fundamental structure, it's a domain interaction between arginine 61 and glutamine 255. And this is work by the late Carl Geisgraber at UCSF that has elegantly demonstrated this domain interaction that differs between E3 and E4. And this domain interaction is between arginine 61 and glutamate 255. The presence of a positive charge at arginine 112, which defines APOE4, allows this interaction to happen, predisposing this particle to form aggregates. So under the electron microscope, you can clearly see, this is E2, this is E3, and this is E4, that you can clearly see that APOE4 forms aggregates. Now, you've all heard about A beta aggregates, you've all heard about tau aggregates, but less is known about APOE aggregates, and APOE aggregates are believed to form the nidus of the A beta aggregates, allowing A beta aggregates to form into plaques. And later on, patients develop tau aggregation and phosphorylation. My lab has been studying APOE aggregation for the past 4 or 5 years, and we showed that lipidating APOE4 to make APOE HDL particles significantly reduces its ability to aggregate. So one aspect to deal with this aggregation problem is to lipidate APOE by activating a transporter known as ABCA1. So this slide summarizes a lot of the basic science that I'm gonna go over with in the next 10 or 15 minutes. But APOE recycles in and out of the cell to make APOE HDL. And in this recycling process, multiple transporters are affected, including ABCA1, which controls APOE HDL formation, MFSD2 alpha, which captures lysophosphoryl DHA into the brain, CD36 exchanges fatty acids, GLUT1 at the blood-brain barrier to take in glucose, and MCT1 for ketone bodies. Importantly, uh, APOE recycling also controls LRP1, which affects the MMP9 enzymatic digestion of the blood-brain barrier, and uh, the cycling of APOE controls the MIPK, PPAR gamma, PGC1 alpha transcription system, which is critical to how the body responds to dietary interventions. So as APOE4 aggregates in the lower panel, it affects the transport through a lot of these transporters, affects the signaling by the MAPK, PGC1 alpha, PPAR gamma, and disrupts the blood-brain barrier. These changes predispose the aging brain to inflammation and oxidative stress. So recently, my colleague at USC, Betsy Zlokovic and Axel Montan published in Nature a paper showing that APOE4 carriers, before they develop cognitive decline, they show defects in the blood-brain barrier, as evidenced by imaging and by CSF studies. I would like to highlight to this audience that the blood-brain barrier not only controls the transport of proteins and other factors into the brain, but also controls how nutrients are transported into the brain. In the blood-brain barrier, GLUT1 is critical for the transport of glucose into the brain. MFSD alpha 2 alpha controls the transport of um, DHA into the brain, and MCT1 factors into the transport of ketone bodies. And it's important to realize that APOE4 carriers, when they have defects in the blood-brain barrier, they also have defects in nutrient-brain transport. So let's start by glucose. You might have heard or you might know that APOE4 carriers have defects in glucose transport into the brain. One of the largest study was the Mayo Clinic aging study, where they looked at almost 900 individuals and examined FDG PET scans in this cohort. In a nutshell, they found that APOE4 carriers above the age of 60 start showing lower brain glucose uptake, and that this lower glucose brain uptake is accentuated when they have amyloid deposition, as demonstrated by PIP positivity. So again, what this slide shows, that older APOE4 carriers have defects in their ability to transport glucose into the brain, and that is accentuated by amyloid pathology. And since glucose transport by FTG-PET is a GLUT1 mediated glucose uptake, this is not regulated by insulin and not regulated by factors that control the expression of the insulin receptors, such as exercise or diet. And this is more related to the pathology that's intrinsic to Alzheimer's disease. We have shown that older APOE4 carriers with greater amyloid deposition in the setting of mild AD is associated with lower transport of DHA to the brain. So in the ADCS-sponsored clinical trial, APOE4 carriers had less increase in CSF DHA compared to non-carriers. In addition, having lower CSF A beta 42, which is an indirect surrogate marker of brain amyloid deposition, was associated with lower DHA transfer in the CSF. So not only glucose is less able to get into the brain in older APOE4 carriers with amyloid pathology, but also omega-3s such as DHA. Now this translated or explained why in this trial, APOE4 carriers showed a differential response to omega-3 supplementation. In this trial, APOE4 carriers had no response, while non-carriers may have had some response, although this is a post hoc analysis and we have to take these findings with a grain of salt. So insights so far is older APOE4 carriers with mild AD or greater amyloid pathology have a lower brain transport of glucose and omega-3s. But why are APOE4 carriers with AD resistant to supplementation? So I would like to entertain this hypothesis that is it possible that the brain, in light of less omega-3 and less glucose transport, is auto-digesting or eating itself to get ATP to maintain its neuronal firing. So the question that I'm raising right now, is it possible that the APOE4 brain is starting to utilize its own fat as a source of fuel? So let's get a little bit more hints into what's happening. So we examined in detail what happened in the Quinn trial, or the ADCS-sponsored DHA trial, and we found that the DHA to arachidonic acid ratio differed by genotype after supplementation. So if you compare the E4 homozygotes to the E3 homozygotes, you could clearly see a drop in the DHA to AA ratio. That also held true for EPA to AA ratio. Now, this could be a biomarker, but more importantly, this is a marker of something else. A greater arachidonic acid to DHA ratio suggests that an enzyme is activated, and this is the calcium-dependent phospholipase A2. The reason why this enzyme is important because it maintains how much of those fatty acids are stuck to the membranes of neurons in the brain. And it also regulates how astrocytes and other glial cells are sensing fatty acids as they are transported from the blood into the brain. Just a reminder that this system is regulated by the MIPK P38 signaling system. And we already know that accumulations of A-beta oligomers can activate CPLA2, and knocking out CPLA2 in AD mouse models rescue the behavioral changes observed in these models. So we wrote a review in 2017 hypothesizing that APOE4 may actually activate CPLA2. And I can tell you that most of my hypotheses are wrong. You know, a lot of times I realize how little I know, but this one did not turn out to be wrong for some reason. So we hypothesized that APOE4 could increase CPLA2 activity, and that could be driving neuroinflammation and oxidative stress. So to answer this question, we looked at the MAPK P38 CPLA2 signaling activated with APOE4 using human APOE knock-in mice, primary astrocytes, and human brains. This work has been led by an outstanding postdoc in my group, Xiaowei Wang, and he takes all the credit for this work that I'm going to present. We were also fortunate to collaborate with David Bennett and Zoe. I can't pronounce her last name. Zoe, please forgive me if you're hearing the talk. From Rush ADRC brain repository. So that we had access to these AD and NCI, no cognitive impairment brain samples that differed by genotype. And we also had access to a hippocampus from the USC ADRC as well. So what we found is that P38 MIPK phosphorylation was greater in APOE4 astrocytes, the lower panel, and greater in APOE4 mouse cortex. So this is the primary astrocytes, and this is the mouse cortex, but not the ERK MAPK signaling pathway. So we also looked at CPLA2 activation and its cascade, and this is CPLA2 phosphorylation and total CPLA2 in primary astrocytes. This is leukotriene B4, which is a downward signaling cascade from arachidonic acid metabolism. This is arachidonic acid efflux, and you could see that not only CPLA2 is phosphorylated, arachidonic acid is more transported and converted into leukotriene B4. Importantly, when we inhibited CPLA2, we lowered leukotriene B4 levels in APOE4 models compared to APOE3. So we also found this phenotype in APOE targeted replacement mice. At 8 months of age, we were observing increased phosphorylation of CPLA2 in APOE4 TR mice compared to APOE3 TR mice. So let's talk about human findings now. So looking at the human cortex ROSMAP Rush ADRC database, we found that APOE4 no cognitive impairment group, which had lower amyloid pathology at autopsy, there was no difference in p38 phosphorylation. In contrast, E3, E4 carriers with amyloid pathology at autopsy, significant amyloid pathology, and dementia had a significantly higher of p38 phosphorylation. This means that it's not just the genotype that enhances the phosphorylation of p38, but also the disease stage. In agreement, we found that phosphorylated CPLA2 is not statistically, although there was a trend toward higher signal in the E3, E4 no cognitive impairment group compared to the actual E3, E3 no cognitive impairment group, but significantly more CPLA2 phosphorylation in the human brain of E3, E4 carriers compared to the E3, E3 carriers. So to summarize, it appears that APOE4 in AD induces the MAPK P38 activation that can predispose the brain to neuroinflammation. So with AD pathology, APOE4 activates the system that allows extracting arachidonic acid from phospholipids to liberate arachidonic acid, which gets converted to leukotrienes and induces oxidative stress and inflammation. So that actually raises the possibility that CPLA2 inhibition could be a drug target for attenuating APOE4-driven neuroinflammation. So what does it mean that MAPK CPLA2 signaling is activated in E4 carriers with AD? It is possible or plausible that the CPLA2 activation could represent a compensatory mechanism to extract PUFAs from membrane phospholipids as a source of fuel at a time of reduced brain glucose utilization. Is the brain auto-digesting to generate ATP? But also the implication of greater CPLA2 activation is that more omega-6 consumption is going to translate into more neuroinflammation because omega-6, like arachidonic acid, will be liberated at a higher speed by CPLA2, and that leads to more neuroinflammation. And importantly, when you have higher CPLA2 activation, increased omega-3 consumption may not be very effective because those omega-3s are not going to latch to the membranes because they're being deesterified by this enzyme. So then the question is, can omega-3 consumption in APOE4 carriers help before the onset of disease? So there is inconsistent literature in the field, but some studies suggest that it does, others suggest they don't. And let me review this in a nutshell. So this is an example of 4 studies, 3 of which show that a healthy lifestyle, including diet, fatty fish consumption, fruits and vegetables, is associated with reduced dementia risk, but only among APOE4 non-carriers. Note that these are 55 to 65 and older. In contrast, one epidemiological study from Finland showed that moderate intake of PUFAs is associated with decreased risks among APOE4 carriers, but this population is substantially younger, 44 to 60 years of age. So timing does matter at when the intervention. We conducted PET scan DHA studies with Stanley Rappaport and John Amhau at NIH and published that younger APOE4 carriers, unlike older APOE4 carriers with AD, have an increase in brain DHA uptake, suggesting a deficit in the brain. And the brain is using so much omega-3s, but it's able to compensate by sucking all the omega-3s from blood and maintaining cognitive function. One of the advantages of 11C-DHA PET scanning is that they allow you to assess the turnover of DHA. And John Amhau has published in 2013 that the estimated turnover of DHA is around 2.5 years. This is important and has implications to clinical trials, because if it takes so long to remodel DHA into the brain, we shouldn't be expecting that a trial between 6 months and 3 years will lead to significant replenishment or change of DHA within neuronal membranes. So who are the individuals that could benefit from PUFA supplementation? And let me give you a hint here, or a spoiler alert. Not everybody will benefit from PUFA supplementation. So we have published a small paper, or a paper in a small subset of the aging brain study, suggesting that people at the lowest quartile of DHA levels in blood had a significantly higher amyloid deposition index by PET-PIB. At the same time, Zhang et al. Did a massive meta-analysis involving 181,000 people and found that people who don't consume any fish or they have less than one serving of fatty fish per week have an increased risk of Alzheimer's disease. But I wanted to use these rectangles to show you that the relationship is not linear. And I know that Cecilia's group has published a similar paper in Alzheimer and dementia, so supporting this concept that the greatest risk of Alzheimer's disease happens between people who don't eat fish or have one fatty fish consumption or less. As you increase the amount of fish consumption, the actual benefit gets attenuated. So then there are some still unanswered questions. What is the optimal DHA supplement dose? Would APOE4 carriers benefit if we started before the onset of cognitive decline? And do they have a brain delivery defect even before the onset of cognitive decline? So to address these questions, we conducted from 2015 till 2018, the DHA brain delivery pilot trial. And this trial was just published in July in eBiomedicine. So this trial was only 6 months. It was a small trial, relatively small, with only 33 people randomized to high-dose DHA, defined as 2 grams per day, versus placebo. Everybody got a high-dose vitamin B to make sure that we are incorporating DHA into phospholipids. But the trial was difficult because we had to screen 2,000 individuals to identify who is willing to do CSF lumbar punctures before and after treatment to assess the primary outcome, which is the change in CSF DHA. In addition, to enter into this trial, your DHA intake had to be less than 200 milligrams per day, which was equivalent to less than 2 servings of fatty fish per month. So this is the baseline characteristics. And again, these are the mean age was 69 and the MOCA was 27 or 28. And this is already published if you want more information in eBiomedicine in July. What we found in a nutshell is that 2 grams of DHA per day translated into a modest 28% increase in CSF DHA, as opposed to almost 250% increase in plasma DHA. So the bottom line is that changes in plasma do not particularly or do not exactly reflect changes in CSF. And APOE4 carriers had slightly less increase in DHA and CSF compared to non-carriers, which suggests that APOE4 reduces omega-3 brain delivery before the onset of cognitive impairment. So again, those matters. Despite a large increase in plasma DHA, there was a modest increase in CSF DHA. And this may probably provide some insights on why past studies that used less than 1 gram of DHA per day may not have translated into cognitive benefit. With lower DHA doses, it's likely that the brain bioavailability is limited. So APOE4 carriers with greater amyloid pathology had a lower increase in total CSF and DHA and EPA after supplementation. The implication is that APOE4 carriers may require longer duration of high-dose DHA supplementation, and that reduced DHA delivery precedes cognitive impairment. So the obvious question, is there a window for slowing cognitive decline in APOE4 carriers with limited fish intake by high-dose DHA supplementation? So let's shift gear a little bit into recent clinical trials. I do have to put a disclosure here though. The example presented here have low effect sizes. In clinical trials with relatively large sample sizes, low effect sizes could be observed by chance alone. So that's a disclaimer that the findings that I'm going to discuss could have happened by chance. Let's start by the multi-domain Alzheimer prevention trial, or the MAPT trial. This was a 3-year interventional trial which looked at cognitive training, advice on nutrition, physical activity, combined or in isolation with PUFAs versus placebo. The inclusion criteria included subjective cognitive decline and frailty, The mean age was 75, MMSE at baseline was 28. The PUFA dose were 800 milligrams of DHA per day and 250 milligrams of EPA per day. The primary composite outcome was not met. The trial was negative. And I understand Nikos presented a deeper discussion on this trial in our previous webinar, and I would urge you to look at the previous webinar to get an in-depth understanding of what happened with these trials. But for the sake of this talk, I'm going to zoom in into APOE4. In the subgroup analysis in this trial, APOE4 carrier, lower omega-3 intake, and high cardiovascular risk factor score may have better responded to the intervention than non-carriers. Let's turn a little bit now to the Finnish geriatric intervention study, or the FINGER trial. This is older adults with cardiovascular risk factors. The mean age was 70. The MMSE at baseline was 27. The intervention included diet, exercise, cognitive training, and vascular risk monitoring. In the supplement, the paper shows that after the intervention, exercise levels and fatty fish consumptions were the only two statistically significant changes in the treatment arm. The outcomes in this trial, unlike MAPT, were met. So there was a statistically significant difference in the NTB total score that reflected executive functioning and processing speed, but not memory. However, it's important to realize that the Cohen's effect size was 0.13. This is a very weak effect size. And this finding could have happened by chance. So in the subgroup analysis of this trial, APOE4 carriers may have had a better response to the intervention than non-carriers. So if you look at the Solomon et al. JAMA Neurology Review in 2018, the intervention appeared to be at least 2 times more effective in E4 carriers than non-carriers, but the statistical interaction term was not significant. So finally, I'm just going to talk a little bit about the Lipidi diet. Lipidi diet is an initial paper was a 2-year intervention using Fortis and Connect, which contains about a gram or a little bit more than a gram of omega-3s combined with vitamin B, uridine, and choline. In this trial, approximately 150 people were randomized to this Fortis and Connect versus a placebo control. And people were followed for an average of 24 months. And I understand that the trial has a 3-year extension that was recently reported at AAIC. It's important to note that 60% of the population of the Lipidi diet were APOE4 carriers. The mean age was 70, baseline MMSE was 26, and they required mild hippocampal atrophy to get into the trial. The trial did not meet its primary outcome. It met some of the secondary outcomes, and there was a higher than expected conversion to Alzheimer's disease dementia, suggesting that these people were on the sicker side. So I wrote an editorial or commentary on the Lancet Neurology about this trial, that this population targeting prodromal AD could have been too late for prevention. And that was supported by a pre-planned analysis suggesting that a lower MMSE score or an MMSE score greater than 26 was associated with a better drug placebo response. So the less severe the disease at baseline, the better the response to this intervention compared to people who had lower MMSE scores and more advanced disease. So that raises the question of whether prodromal AD, as defined by this trial, is too late. So let me summarize, and this would be my last slide. So we know from many studies, and one of the key studies is Caselli et al. In the New England Journal of Medicine 2009, that APOE4 carriers start to have a drop in their cognition after the age of 60, more around 55 in homozygotes and 65 in heterozygotes. So let's assume this is a hypothetical model of APOE4 carriers who are going to develop AD dementia. Not all APOE4 carriers. In fact, the majority of APOE4 carriers will not develop AD dementia. But let's assume this is focused on those APOE4 carriers will develop AD dementia as they age. They go through 3 phases: a normal cognition phase, a pre-AD phase, and I'm avoiding the term prodromal AD because we don't know how to define it yet, and an AD dementia phase. I showed you using PET scanning that during the normal cognition phase, 35-year-old APOE4 carriers had greater brain PUFA utilization, suggesting that they're extracting all the PUFAs they can get from blood to maintain normal cognition. As APOE4 carriers age, APOE4 itself as a protein starts to aggregate. This aggregation precedes amyloid beta deposition, but starts to accelerate between the ages of 60 and 70. During that time, we know that there is blood-brain barrier dysfunction, decreased glucose brain uptake, decreased omega-3 brain uptake, and amyloid accumulation. Now, I showed you today that in patients with Alzheimer's disease, dementia, and APOE4, there is activation of the CPLA2 system that contributes to the loss of omega-3s from the brain. The implication of these observations suggests that, younger APOE4 carriers do not have the leeway to have limited omega-3 intake. They need to have sufficient omega-3 intake to maintain cognition. As they transition into pre-AD, or as they transition to AD, we already know that APOE4 carriers do not respond from omega-3 supplementation, largely because the enzymes are activated that show up omega-3s in the brain. So would high-dose omega-3 supplementation work in the pre-AD phase? And finally, is there a role for CPLA2 inhibition in patients with early AD dementia or late prodromal AD? So to address this question, we were fortunate to receive funding from both the NIA and the Alzheimer's Disease Drug Foundation to run a randomized clinical trial that we call PREVENT-E4. The trial started recruitment in 2019, and it was projected to end unreported in 2024. Unfortunately, because of COVID, we had to halt recruitment in March for 6 months, and we expect now a year in delay in completion of this trial. This trial was designed to answer this simple question: are APOE4 carriers before the onset of cognitive decline or Alzheimer's disease dementia receiving higher doses of DHA, 2 grams per day, as supported by our pilot trial? Would that lead to any improvements in cognitive scores, hippocampal volumes, structural and functional connectivities as secondary outcomes? And do they also have a brain DHA delivery defect? So this trial, although this is not a massive trial, it's stratified by APOE4 specifically to help us understand the APOE4 effect, and will provide interesting observations, support a much larger randomized clinical trial across multi-centered studies to address the question of high doses of omega-3s and cognition in APOE4 carriers. So I want to finalize by acknowledging the many people who have worked on this project with in my lab and our collaborators: Isabella, Victoria, Ashley, Shawei, Carla, Jasmine, Shule, Christiana, Darren, Juno, Kaylin, and everybody else. Helena Chu has been an outstanding mentor from the USC ADRC. Lon Schneider, an outstanding colleague, mentor, and friend. Meredith Brasky for imaging. Lena and Carol for neuropsych testing. The Rush ADRC, Zoe and David Bennett to get the brains. Recent collaborator, Hao Wang, studying the brain CPLA2 activation. Michael Harrington and Alfred Fonte for lipidomics. Wendy Mack Naoko for biostatistics. And most importantly, our funders. I happen to be fortunate to receive funding from the Alzheimer's Association early on in my career. And we got fortunate to get funding from the ADDF and from the National Institute of Aging. So we probably have about 20 minutes for discussing or going into a discussion on this presentation.
Thanks very much, Hussein, for the brilliant presentation and massive amount of work, which is also very original. I think you are one of the single labs in the world doing that kind of work, and it really helps to understand these inconsistent findings we have in of epidemiological studies and clinical trials. So we are going to go into the questions in the Q&R. I'll start with the first one from someone who is anonymous. Do rodents naturally have different forms of APOE?
Rodents have their rodent APOE, and the way to go around that is to create a mouse model where you knock in the human APOE2, APOE3, and APOE4. Now, it's important to realize that the rodent APOE or the murine APOE does not aggregate the same way as the human APOE. And more importantly, murine A beta does not aggregate like human A beta. So the field now is moving toward humanized forms that don't overexpress APOE or A beta as a better model of late onset APOE.
All right. So from Monica Crane, given APOE4 lipoprotein transport changes and possible lower response to statin therapy, do you recommend statins?
I don't think I have the answer to this important question. Although I would say that statins work in patients that are at highest risk of cardiovascular risk factor or having a heart attack or disease. So if somebody has a low risk for having a heart attack or a stroke, they should not be on a statin. If somebody have a high risk of getting a heart attack or a stroke, they should be on a statin because they save lives.
All right. From Claudio Babilone, some researchers think that from a physiological point of view, A-beta-42 is an insulin-like hormone facilitating the intake of glucose into the neurons. So the initial hyperproduction of A-beta in the brain is a beneficial mechanism of homeostasis to compensate the poor intake of glucose into the neurons, something more evident in APOE4 carriers. Can you comment on their thesis?
That's a fascinating observation, and there is a literature supporting that early increases in A-beta accumulation are compensatory changes. But the bigger question is there a possible point where the increase in A-beta accumulation generates a toxic species that could lead to cognitive decline? And I don't think the jury is out yet. The majority of clinical trials have been negative, supporting that we should not be treating A-beta as the holy grail of Alzheimer's disease. But the question regarding A-beta oligomers has not been fully addressed. Although, if you were to ask my own opinion, I think we should be focusing on other non-A-beta research in AD to get a more comprehensive view of how this pathology is happening, particularly lipid metabolism. I know I haven't addressed the insulin signaling aspect, but I don't know enough about that pathway to be beneficial.
All right. From Sumanto Mitra, does this revelation about omega-3 consumption means that vegetarians or vegans have higher susceptibility to develop Alzheimer's disease if they are carrying the APOE4 genotype?
Wonderful question. And I think that question has bugged me for a long time in the middle of the night. Why don't we see an epidemic of Alzheimer's disease among vegetarians? And the answer partly to that question is related to enzymes that can convert alpha-linoleic acid to DHA. That study has not been done yet, but it's plausible that vegetarian can upregulate the desaturases that can make omega-3s from other sources of omega-3s such as nuts. If that's the case, nut consumption can form a pathway to compensate for less consumptions in vegetarians. But not having nuts or fish is a recipe for a disaster for the brain, I think.
Maybe we will see that in 20 years or so. We'll see. Okay, were no females included in this study? I'm not sure about what the study is about.
So our studies have small sample sizes, partly because they're very expensive to do. But as we write more grants and as we follow NIA's or NIH's recommendation, we are increasing sample sizes to accommodate for female sex, both in animal models and in human studies.
Okay, a couple of questions from Christy Pangni. Why did you use algal form of DHA for your trial?
Simply because in the Quinn trial, they used the same DHA at 2 grams per day and found, although found no response, but they found a differential effect by APOE4. That was the rationale in 2015 when we started. However, we know that there are other forms of DHA that might have better BBB transport. However, I have to caution the Christie that all of these hype about all kinds of other DHA formulations have been largely limited to animal studies and not to humans. So we still have a long way to figure out a formulation that can better penetrate the BBB, including krill oil and other forms of DHA.
All right. She also asks, are there any evidence relating dose response between plasma and CSF DHA levels to explain the nature of these relationships?
Yes, greater doses of DHA leads to greater both increases in plasma and CSF. But I have to caution you that the increase in plasma is not exactly proportional to the increase in CSF, especially when you have APOE4 and brain amyloidosis, and there is a break in the transport between those two compartments.
Yeah. Okay. Are you tired, Hussein? So you don't seem to be. So we go on. Susan Gibson asks, she says, excellent presentation, great research. What do you think is the best dose of DHA? What do you think is the best dose of DHA for the general population, regardless of APOE status, since many people don't know their status? Would you change the recommendation based on age from what you know?
Excellent question. The best dose of DHA is 1 serving of salmon per week. I don't recommend supplements, and if I knew that supplements work, I would not be doing my trial. The age recommendation requires a massive multi-center trial. So all of these presentations are at this point suggestive, and I would urge you not to prescribe or make deeper recommendations without good evidence.
All right, we have Caroline Kaufman asking, do you have any plans to study cerebral blood flow changes with DHA supplementation?
I guess the question is, do I have any funding to study cerebral blood flow changes with DHA supplementation? The answer, not now, but hopefully when we get more funding, we would definitely be interested in this effect.
Sabine Krum, are there any studies investigating the impact of nutrition in younger age on cognitive dysfunction in older age? Or is the impact of diet primarily important at age 60 plus in APOE4 carriers?
I tried to convince the audience today that the lifestyle, including diet interventions, are most effective in APOE4 carriers when started during middle age and not above the age of 60. The problem becomes pragmatic. When you choose a population between the ages of 40 and 60 and they don't decline on cognitive scores, the trial is incredibly difficult to do. And that's why we're starting from the age of 55 to 75. But epidemiology studies and our basic science studies suggest that if there's a role for supplementation, it's going to be before the disease develops.
Okay, we have great talk, Dr. Yassine. I may have missed it due to distractions in my lab, but did you find any sex-based differences in brain metabolism or in any of these, sorry, the window is moving while I'm reading, or in any of these presented, especially at some of the time points shown, which may be post-menopause?
That's a wonderful question, Ben. And we published in the Journal of Alzheimer's Disease this year that there is a sex effect on the response to supplementation in females versus males. I did not present this here, but apparently females have a different response to male to DHA supplementation. And I know Anne-Marie Minahan is interested in that topic as well. We also published this year in Neurobiology of Aging that menopause has an effect on the response to weight loss in APOE4 carriers versus non-carriers. And I may not have the time to go over this, but there is an effect of menopause on the relationship to the response to interventions, dietary interventions such as weight loss and possibly omega-3 supplementation.
Okay, great presentation. Would you recommend for APOE4 individuals that are still within the normal cognition stage to take omega-3 supplements that include both EPA and DHA? Is it best to take fish oil supplement instead? If so, how much should be in the supplement?
I would recommend to have one serving at least per week of fatty fish as a source of your omega-3 supplements because the epidemiology is supportive. We don't have clinical trial evidence to support omega-3 supplements at this point. And I would caution that our findings are preliminary and we still need large randomized clinical trials before we make such a recommendation.
Okay, Aaron Smith, you briefly spoke about APOE4 carriers and decreased brain glucose metabolism. Could you touch more on that? Sorry, the question is broad.
Right, so we know that APOE4 carriers and this, work by many people, including the Zlokovich lab, have defects at their blood-brain barrier that appear before cognitive decline. And we know that the glucose uptake in the brain is largely driven by the GLUT1 transporter, which is at the blood-brain barrier that could be affected by AD pathology. So part of the reasons why APOE4 decreases glucose uptake because it damages the transporters that capture glucose into the circulation. This forces APOE4 to find an alternative source of energy. And fat could be that source. And I know Angela Hansen and Lance Johnson are working on brain energy preferences by APOE4.
Mm. I'm afraid we are not going to be able to take all the questions, but we still have a couple of minutes. Do we expect COVID-19 to affect symptoms of Alzheimer's disease?
Yes, we do. And I know NIA has a strong interest in studying this question. I just want to sidetrack and say that APOE4 carriers appear to have worse neuroinflammation when they are infected with COVID-19. And one of the findings that we're studying in CPLA2 activation could be explaining why APOE4 carriers are at increased risk of neuroinflammation.
Okay, we have Nick saying great talk, really gave a great overview, and I am sure we all learned a lot. Do you exclude participants with already existing amyloid pathology from your trial, since you indicated that it may be quite pertinent for omega-3 effects in carriers?
No, we do not, for two reasons. One, there is a timing effect where you may still have some amyloid pathology, but you can still respond to supplementation. Two, it's not practical in a trial to do CSF or PET scans. It's too expensive. But there is a point by which amyloid accumulation, and maybe the oligomers here play a role, activates CPI2. But we know amyloid accumulation can start as early as 40 years of age. So from 40 to 60, there could still be amyloid accumulation and response to omega-3 supplement.
All right. Are you seeing sex by APOE4 effects with your omega-3 research?
We have a paper published in the Journal of Alzheimer's Disease that discusses sex effects, and females respond differently than males. Yes.
Okay, Jean Bowman, nice presentation. Thanks. How will PREVENT-4 trial address baseline variation in plasma omega-3 status as an explanation for brain delivery of omega-3?
That's a great question, Jean. The problem is we don't have the resources to measure or stratify omega-3 levels at baseline. What we do is we just look at how much fish they're consuming and use that as our guide to limit the variation. But we do expect some variation, and I expect the best we can do is statistical modeling to account for this variability.
You're going to measure blood DHA, of course.
Yeah, blood, RBC and plasma, yes.
Okay. If DHA is broken down in APOE4 carriers to provide energy, could this be prevented by providing an alternative source of energy, such as ketones or short-chain fatty acids, in addition to supplementing DHA?
Well, we know from 2 ketone body trials that were negative. APOE4 carriers were non-responsive, and partly because the actual transporters for ketone bodies at the blood-brain barrier are probably also affected by the disease pathology. So yes, there could be a specific diet that can help the aging APOE4 brain, but we still don't know what that diet is.
Mm. Okay, thank you for the great talk. What is the connection between insulin resistance and omega-3 uptake, and how that connection relates to APOE genotype? Thank you.
That's a very complex question, and in a nutshell, there are evidence suggesting that omega-3 intake and combined with weight loss ameliorates insulin resistance as opposed to the individual intervention alone.
Does a ketogenic diet that appears to help in MCI population have any benefits? I think you already answered. Do you want to complement?
No.
So Nick, on a different note, although not directly to your talk, could you say something on the interaction of omega-3 efficacy in relation to the B12 homocysteine status?
That's another question that has not been fully resolved. As we see with our AAIC NMDP abstract presentations, we got discrepant findings. But in a nutshell, you need adequate vitamin B levels to be able to incorporate omega-3s into phospholipids. How much do you need? At what time should this intervention happen? We still don't know. And to avoid having a vitamin B confounding effect, in our trial, we give all participants, whether placebo or DHA, a vitamin B complex.
All right. Okay. Is there a role for downstream DHA products, protectin and resolvins, that will compete with LTB4 to resolve inflammation?
Yes, there is a role for downstream DHA, and that's why DHA consumption can replace arachidonic acid signaling. However, there is a timing effect, and at some point, the DHA consumption may cease to work.
All right. I think maybe it's time to stop. What do you think, Hussein? Do you want a couple of more questions?
Maybe a couple of more, and then I can take my break.
Okay, so last one. Is there an evolutionary advantage for E4?
That's a wonderful question, and I urge you to read the review I wrote with Tuck Finch, who has a deep interest and understanding of the evolutionary benefit of why so many people carry the APOE4. And the short answer is yes, in populations such as Nigeria, where there's a lot of parasitic infections, APOE4 carriers outperform non-carriers in birth weights, in deliveries, in cognition, and almost everything. Although, again, you have to take these findings with a grain of salt. But it appears that we have mishandled the environment. APOE4 carriers mishandles the aseptic environment and auto-attacks itself.
Wow, okay. Wow, you did a great job with answering all these questions. That's crazy. Thank you so much. I don't know if you are going to join us for the concluding remarks, Heather and Jody.
I think the only thing that we'll share, and we had put the link in the Q&A, is that this will be posted in 2 weeks on the association's website, both through iStart, but then also under the research webinars. So we encourage you to take a look. I think there were a couple of questions that we, of course, didn't get to. We got to as many as we can. Huge record number, I think, through like 40 questions. There were definitely a lot of questions around sex differences and on other potential contributors that may intersect or interact with nutrition and diet and in particular DHA. So huge thank you, Hussein, for the talk. It was, I think, a really excellent overview of both your work, but also the field and where things are going. And stay tuned. I think we've got a few future webinars scheduled in the near future. And so maybe concluding comments to you, Hussein and Cecilia, as the Nutrition, Metabolism, and Dementia Professional Interest Area Chairs.
I will let Hussein conclude his seminar.
No, I would just want to conclude by asking people on the call to join the NMDP. This organization is really made for serving you as a junior investigator. It gives you the opportunity to present your work, get awards. And although Cecilia and our term is going to be ending up soon, I think we need more nutrition researchers. We are going to have a symposium in July, the date to be finalized soon, about nutrition and the brain. And we hope that you all join us for that symposium.
Great, thank you so much, Hussein, for this amazing seminar, and thanks all for joining.
Thank you.
And we'll see again in early 2021 for the next one, I think.
Excellent.
Thank you. Bye. Bye, everyone.